हेलो डियर स्टूडेंट्स, इम्यूनोलॉजी के नेक्स्ट टॉपिक में हम लोग मोनोक्लोनल एंटीबॉडी टॉपिक डिस्कस करने जा रहे हैं। दिस इज़ वेरी इंपॉर्टेंट टॉपिक फॉर द नीट पॉइंट ऑफ व्यू। मोनोक्लोनल एंटीबॉडी दैट यू कैन राइट इन दिस वे, स्मॉल m कैपिटल A स्मॉल b, और यू कैन राइट स्मॉल m o एंड देन AB। इस तरह से भी MCQ में कई बार पूछ लिया जाता है कि हाउ कैन यू राइट मोनोक्लोनल एंटीबॉडी। सो दिस इज द एक्सप्रेशन ऑफ मोनोक्लोनल एंटीबॉडी। Before going to start this topic, I would like to discuss with you the importance of antibody. You know, when particular antigen is injected into human body or any other animal or organism produces particular antibody to that particular antigen. That's why this is called as monoclonal antibody. अगर हमारी बॉडी में कोई एंटीजन इंजेक्ट किया जाएगा तो उस एंटीजन के अगेंस्ट में जो एंटीबॉडी डेवलप होती है उसे हम और आप कहते हैं मोनोक्लोनल एंटीबॉडी। सपोज आपको किसी डिजीज़ के अगेंस्ट में एंटीबॉडी डेवलप करनी है, जिस डिजीज़ के अगेंस्ट में डेवलप करनी है वो डिजीज़ किसी पैथोजन से हो रहा होगा। उस पैथोजन को हम लोगों ने रैट में इंजेक्ट कर दिया। We are writing antigen. You know how can you write antigen? Capital A small g. We are injecting antigen into a rat. The lymphoid organ of rat will start forming antibody to that particular antigen. Here what are we doing? Spleen, you know, this is the example of lymphoid organ. We will separate the spleen from the body of rat. Spleen is capable to form particular antibody to that particular antigen that you have injected into the mice body. But there is a drawback. Spleen is capable to produce antibody, but the lifespan of spleen is limited. So the next option that we are going to see here, we are taking myeloma cell. This is the cancerous cell. This is the cancerous lymphocyte cell. It has the property of uncontrolled division. Myeloma cell can uncontrolled divide. What are we doing? We are fusing myeloma cell with spleen cell. Myeloma cell has the property of uncontrolled division. Spleen cell has the property to produce antibody to particular antigen. When both of these two cells fuse, it forms hybridoma cell. When you are fusing two different types of cell, this is called hybridoma cell, and this technology can be called as hybridoma technology. Hybridoma technology. So if someone asks what is hybridoma technology, you are fusing two different type of cell. You may ask, how can we fuse two different type of cell? There are some agents that we use in biotechnology. Electric shock by giving electrical stimulation, polyethylene glycol PEG, Sendai virus. These are the agents that we can use to fuse two different type of cell. And when they fuse, they form hybridoma cell. Hybridoma cell, it has the property to produce antibody against the particular antigen, and hybridoma cell has also the property of uncontrolled division. But when you are getting hybridoma cell, you are getting actually different type of hybrid cell. Here I have shown with different color pen. You can see black, red, blue, green. We are getting different types of cell. All these different types of cell, suppose this is the black cell, it will produce antibody. Red cell will produce some different antibody. All cells are capable to produce antibody. But what is our necessity? What is our requirement? We need particular antibody. That antibody should be against that particular antigen. Remember here, all these cells are not capable to produce particular antibody to that particular antigen. All cells are capable to produce antibody because all of these cells are developed from spleen. all of them have the property of uncontrolled division, but only few cells are capable to produce particular antibody. The cell which is capable to produce antibody against that particular antigen, we will separate that cell, we will culture that cell. How can we separate? We can separate by the use of HAT medium. HAT medium का इस्तेमाल करके उस particular cell को we can differentiate which is capable to produce antibody against a particular antigen. What is HAT medium? Here I have written the full form of HAT: hypoxanthine aminopterin thymidine. Hypoxanthine aminopterin thymidine. This is the full form of HAT. This HAT medium is used to separate the particular hybrid cell which is producing an antibody against a particular antigen. How does it work? What is the significance of it? This is not at all important for us. We just need to remember we use the HAT medium to separate that particular cell which is capable to produce antibody against a particular antigen. Okay, this is the culture medium. This is the culture medium. In this culture medium, we have separated that particular cell. That particular cell will grow to form particular antibody. Then we are doing the subculturing. Subculturing means a large culture media has been divided into several small culture media so that it can produce required amount of antibody. So all the red cells that you are seeing here, all these cells actually will form what? Actually they will form or release antibody. They will form an antibody. So this is called as monoclonal antibody, magic bullet. This is called as magic bullet. देखो कितना नेम कितना सिग्निफिकेंट है इसका. एंटीजन को टारगेट करके उसी एंटीजन को किल करेगा. आज के प्रेजेंट सिनेरियो में सोचो कितना सिग्निफिकेंस है. यू नो आपकी बॉडी में वायरस का, बैक्टीरिया का अटैक हो गया है. आप उसी बैक्टीरिया वायरस को टारगेट करने के लिए पर्टिकुलर एंटीबॉडी प्रोड्यूस कर रहे हो. दिस टेक्निक वाज फर्स्ट टाइम यूज्ड बाय कोहलर एंड माइलस्टीन इन 1975. कोहलर एंड माइलस्टीन इन 1975 दिस टेक्निक वाज यूज्ड बाय द साइंटिस्ट. तो ये कुछ पॉइंट्स हैं इंपॉर्टेंट दैट यू मस्ट नो अबाउट द मोनोक्लोनल एंटीबॉडी. सपोज इन MCQ somewhere is written polyclonal antibody. What is the mean of polyclonal antibody? कमेंट सेक्शन में लिख करके मुझे बताइए या व्हाट्सएप मुझे करिएगा. थैंक यू फॉर वॉचिंग मी.